Today's topic is gram-negative anaerobes. Anaerobic bacteria are of two types. One is spore forming and another one is the non-spore forming. Spore forming bacteria, they are gram positive, and the example for them are Clostridium bacillus. Whereas non-spore forming bacteria, anaerobic non-spore forming bacteria may be of two type. It may be gram positive or as well as it may be gram negative. Gram positive, again, gram positive non-spore forming anaerobic bacteria may be of cocci shape or it may be of rod shape. Whereas the gram negative anaerobic non-spore forming anaerobic bacteria are rod shape, which today we will deal. And examples for these are Fusobacterium, Dichrobacter, Proboscis, Bacteroides, etc. And these anaerobic bacteria generally they exist in the environment and are commensal on the mucous membrane of the animals and the human. Particularly, they reside in the intestinal tract as a normal flora, and frequently these are associated with the opportunistic infections and gets excreted in the feces and can survive for the environment for the short period, and generally the presence of the aerobic and the facultative anaerobic bacteria and in the surroundings, they reduce the local oxidation reduction potential and they make the environment suitable for the survival of and the growth of the anaerobic bacteria. And these may be associated with the different types of the disease conditions like the soft tissue abscess, post-operative wound infections, periodontal abscess, lung and liver abscess, mastitis, foot rot, aspiration pneumonia, peritonitis, pleuritis, myometritis, osteomyelitis, and so many conditions. And the pathogenic anaerobic non-spore forming gram-negative bacteria, these were first recognized by the Leffler in 1884 as an important pathogen in the animals, and Spermans identified these as opportunistic in the humans in the 1890s. These anaerobic gram-negative non-spore forming bacteria, they belong to the different genus and they can be grouped as Bacteroides fragilis group, in which they belong to Bacteroides C family and the genus is the Bacteroides. Fusobacterium species family is the Fusobacteriaceae and the genus is the Fusobacterium. Earlier these belong to the family Bacteroideaceae. Porphyromonas, these belong to the family Porphyromonidaceae and the genus is Porphyromonas, and these also earlier belong to the genus Bacteroides. Prevotella species and the family is the Prevotellaceae and the genus is the Proboscis, and these earlier belong to the genus Bacteroides also. Dichrobacter nodosus, the old name for this bacteria was the Bacteroides nodosus, and this belongs to the family Cardiobacteriaceae and the genus is Dichrobacter. So how we can differentiate these anaerobic gram-negative non-spore forming bacteria? By the gram staining and the morphology, rod shape, cocci, filaments, spiral. These are the different forms in which these bacteria may be present, but the rod form is the most common. And the rods of the Dichrobacter nodosus are thick, straight, and slightly curved, and up to the 6 micrometer long, and they bulge at one or both ends. Whereas the Fusobacterium necrophorum, these have the irregular staining, and these are long, non-branching filamentous form. Coming to the colonial morphology, the colony of the gram-negative anaerobic bacteria, these have a fetid or putrid odor due to the volatile fatty acid production, and the appearance of the Dichrobacter nodosus colony is the variable. Whereas the Fusobacterium necrophorum, these are the gray, round or shiny, and some may be molate. By the differentiation of the motility, see here. In this picture, you can see these are the straight of the curve or the curved rod of the Diclobacter nodosus, and you can find characteristic swelling at the one or the both ends. Here you can see at one end, whereas here you can see swelling at the both ends. And in this picture, you can see the Fusobacterium necrophorum, which is slender, non-branching filament, and which tends to stain irregularly. Pigment production, by the production of the pigment, different genus of the anaerobic gram-negative bacteria can be differentiated. Genus Porphyromonas, these produces porphyrin pigments, and many Probitella species also, they produce the pigments. Fluorescence under the long wavelength UV lights, the Porphyromonas and the Probitella, they give orange to brick red color of the fluorescence, whereas the Fusobacterium, they give yellow is green color the fluorescence, and the Bacteroides species generally they do not fluoresce. And these can be differentiated on the basis of the special potencies, antimicrobial disk susceptibility, fatty acid analysis, and the presence of the isobutyric, butyric, and the succinic acid can be done. MALDI-TOF and the sequencing method are also used for the differentiation. Coming to the virulence factors, these bacteria are also having virulence factors. These contain some cell structures like capsule, fimbriae, flagella, agglutinin, adhesin, LPS, and these play a role in the virulence of the bacteria. Bacillus fragilis, Bacteroides fragilis, capsule of these bacteria, and the pigmented Probitella and the Porphyromonas, these can incite an intense inflammatory response in the host. And the capsular polysaccharide of the Bacteroides fragilis, they can produce abscess even in the absence of the live cells. Some of these bacteria, they produce exotoxins like hemolysin and the leukotoxin, and some also produce extracellular enzymes like neuraminidase, proteases, DNase, and the lipases, which facilitate adherence, colonization, invasion, and the tissue destruction of the cells. Tissue destruction in the host, and some of these bacteria also produce some acid like lactic acid, butyric acid, and also the ammonia, and these are having inflammatory and the cytotoxic effect, and these contribute to the pathogenesis of the bacteria. Generally, these bacteria exert pathogenic effect when the anatomical barriers are breached and the underlying tissue are invaded, but they replicate only when there is the low or negative reduction potential. And these bacteria produce the superoxide dismutase, which provide the ability to survive in the oxygenated tissue until the redox potential reaches the level favoring their growth. And the tissue trauma and the necrosis followed by the multiplication of the facultative anaerobic bacteria, they lower the pH, means the oxidation reduction potential, they lower, and then they favor the condition for the growth of these anaerobic bacteria. And these anaerobic bacteria, they grow in the tissue, and their growth is characterized by the abscess formation and the tissue destruction. And generally, they are found in the mixed type of the infections. And these anaerobic bacteria, they also act synergistically, means two or more bacteria they are helpful for the teacher, like the Fusobacterium necrophorum and the tissue damage facilitated by these facilitate the invasion by the Diclobacter nodosus, and the Diclobacter nodosus in turn provide the growth factor which helps the Fusobacterium necrophorum. Similarly, leukotoxin produced by the Fusobacterium necrophorum, they facilitate the survival of the Tripurella pyogenes, and the in additionally, Tripurella pyogenes, they provide the growth factor which helps in the survival of the Fusobacterium necrophorum. These bacteria are responsible for the production of a variety of disease conditions, and as I told that they are mostly present in the mixed infection. The Diclobacter nodosus, they are involved along with the Fusobacterium necrophorum, Tripurella pyogenes, and some other spirochetes, and they cause the contagious foot rot condition in the sheep. Similarly, Diclobacter nodosus, they can also cause disease in the goat, cattle, pig, and they causes the infection of the interdistal skins. Other disease conditions, Bacteroides fragilis, they can cause disease in the calves, lambs, foals, pigs, and the cattle. And the Fusobacterium necrophorum, they can also cause disease along with the Tripurella pyogenes in the different hosts like the cattle, sheep, pigs, horses, and chickens, and the rabbits. And you can go through the slides to know the diseases. These can be diagnosed by the taking the samples and from the discharges and the lesions, and by the suprapubic puncture in the case of the urinary infections. Samples should be processed promptly after the collections, and the commercial kits and the transport media are also available for the easy collection of the samples and the transportation. Generally, the sample is taken as a tissue specimen over the 2 centimeter cube, where the anaerobic microenvironment is maintained, and if a fluid sample is required, then it can be collected in the syringe. When the air is expelled from the syringe and the needle is plugged, then the anaerobic condition is maintained and is suitable for the anaerobic culture. Direct examination can be done with the gram staining, and in some cases directly the dilute carbamazepine for the 4 to 8 minute staining is done, and the isolation can be done on the enriched blood agar, to which 5 to 10% ruminant blood cells, red blood cells are added, and this can be supplemented. This should be supplemented with the yeast extract, vitamin K, hemine, and media can be used as a selective media with the antibiotics. Apart from this, cooked meat media, meat broth, thioglycolate medium supplemented with the vitamin K and the hemine for the subculture are used, and these media should be pre-reduced to expel any oxygen present in the media. Anaerobic culture can be done with the using the anaerobic jar with the atmosphere of the hydrogen and the 10% CO2. Incubation can be done, should be done at 37 degrees centigrade up to the 7 days, and the selective media are used for the Diclobacter nodosus, and to which powdered bovine hoof is added. The biochemical test can be done. Gas liquid chromatographic analysis is used to detect the volatile fatty acid and the non-volatile fermentation product, which is specific for the each species, and the PCR can be used for the molecular diagnosis. Coming to the treatment, control, and the prevention, drainage of the abscess should be done, and the antimicrobial agent should be done, and penicillin, ampicillin, amoxicillin, chloramphenicol, tetracycline, metronidazole, clindamycin. These are some of the antibiotics which can be used for the treatment purpose. And all the obligate anaerobic are resistant to the all of the aminoglycoside antimicrobial agents, and also to the most of the fluoroquinolones. So these should not be used for the treatment purpose. And antimicrobial therapy should be aimed to the treatment of the facultative and the obligate anaerobic bacteria both, as most of the cases are for mixed infection. And the control and prevention includes the repeated examination, vaccination if the label, treatment of the active cases, and the segregation or the isolation of the active cases or infected animals from the healthy flock. Coming to the next, to the genus wise, so the genus Fusobacterium. This is a Latin word which means a spindle, and but all the Fusobacterium species do not have a spindle-shaped cells. And the common feature of this genus is the production of butyric acid as a major fermentation product. These are the gram-negative bacteria, and these are susceptible for the gram antibiotics, which are generally known for the gram-positive bacteria like the penicillin, tylosin, and virginiamycin. And different Fusobacterium species are there. Of them, Fusobacterium necrophorum is the main species, which is pathogenic for the human as well as the animals. Apart from this, Fusobacterium canifelinum is pathogenic for the canine. Fusobacterium russet, they are for the feline, and other species are Fusobacterium equinum, Fusobacterium necrogens, and the Fusobacterium simum. Fusobacterium necrophorum is the major pathogen, and these are present in the oral cavities, gastrointestinal tract, and genital tract of the animals and the humans. And these are associated with the necrotic lesions in the humans and the animals, and especially in the human, they are responsible for the pharyngitis, especially in the young adults, and these are associated with the Lemierre syndrome primarily in young and healthy animals. In the case of the animals, these are considered as a major bovine pathogen, and these causes the abscess in the abdominal area and can be isolated from the abscess in the abdominal area and the respiratory tract infection of the animals. Fusobacterium necrophorum, there are two subspecies, Fusobacterium necrophorum subspecies necrophorum and the Fusobacterium necrophorum subspecies fundelliforme. Fusobacterium necrophorum subspecies necrophorum is having a more hemolytic activity and is more virulent than the other subspecies. And the Fusobacterium equinum, these are responsible for the production of the oral, paraoral, and the necrotizing pneumonic infection in the case of horses. These bacteria, Fusobacterium necrophorum, these produces a wide range of the virulence factors, and one of them is the leukotoxin. Other are hemagglutinin, hemolytic dermotoxin, other enzymes, extracellular enzyme, lipopolysaccharide, and these all are responsible for the production of the luminal lesions. The most important virulence factor is the leukotoxin, and others are the hemagglutinin and the LPS. These hemagglutinins act as an adhesive and promote the adherence to the and the invasion of the luminal epithelial cells. Leukotoxin, these are active against the luminal neutrophils, particularly they act against the macrophages and the hepatocytes, whereas the endotoxin from the LPS, these are important in the genesis of the hepatic abscess and they produce intense neutrophilia. These are the some of the important diseases which are associated with the Fusobacterium necrophorum, like diphtheria, postpartum metritis, hepatic abscess, black spot of the teeth. In the case of horses, thrush, necrobasmosis of the lower limb, and in the case of pigs, they cause the bulge. These are also associated with the mixed infection, um, like interdigital dermatitis in the case of the sheep, heel abscess, foot rot. In the cattle also, they causes the interdigital necrobacillosis. In the pig also, they are responsible for the foot abscess. Calf diphtheria, this is also known as the necrotic pharyngitis or laryngitis, and generally it is present in the calves up to the age of 3 months. And these can enter through the abrasion of the mucosa of the pharynx or the larynx, and also some by the ingestion of the coarse feed, in the rough feed. And the clinical symptoms which will be seen is are the fever, depression, anorexia, excessive salivation, respiratory distress, and the foul smell from the mouth. And the untreated calves, if the treatment is not done, then they can develop a fatal necrotizing pneumonia. And the treatment includes with the potentiated sulfonamides or the tetracycline in the early case of the disease, where the treatment is effective. Liver abscess or hepatic necrobacillosis. In all types of the cattle and the different parts of the world, all ages are susceptible for the disease. And the secondary to the primary foci of the infection is the ruminal valve, and this disease is also known as the ruminitis liver abscess complex. What happens? The rapid fermentation of the grain by the ruminal microbes, they are responsible for the accumulation of the organic acid, which causes the ruminal acidosis. And when the acid-induced ruminitis is present, it causes the damage of the protective surface, and also this condition can be aggravated by the foreign object like the sharp feed particles and the hair, and this causes the predisposes the ruminal wall to invasion and the colonization by the Fusobacterium necrophylla. So the ruminal wall abscess results, and it shed the bacterial emboli into the portal circulation. And the bacteria from the portal circulation, it goes to the liver and results in the infection and the abscess formation. So sometimes what happens, the abscess gets well encapsulated and having the thick fibrotic walls. And in this case, Fusobacterium necrophorum subspecies necrophorum is more frequently present in the liver abscesses. And also the Trupperella pyogenes is the second most important pathogen which is present along with the Fusobacterium, and it produces a synergistic effect between the Trupperella pyogenes and the Fusobacterium necrophorum. See here you can see high grain feeding, it will result in the acidosis which will cause ruminitis. So bacteria, Fusobacterium will attack and they late and they will cause the bacterial emboli. Through the portal blood, they will enter to the liver and there they will cause the abscess. Here you can see the liver abscess present due to the Fusobacterium. So generally the lesions will be present in the liver, but it will rarely show the clinical signs, and only at the time of the slaughter the lesions can be detected. But it will causes the production losses in the terms of reduced weight gain and the feed conversion efficiency. So in the management aspect, in the feed lot should be aimed to reduce the incidence of the ruminitis. For this, the bulk management should be the proper, means the more grain feeds should not be given to the flock. And the use of the antimicrobial feed additives are also practiced. Leukotoxide based inactivated leukotoxin vaccine are used for some of the parts. Necrotic rhinitis, infection with the Fusobacterium necrophorum. It is also present along with some other anaerobics, and it is a sporadic condition affecting the young pigs, and it is characterized by the suppuration and the necrosis of the snout. And generally through the abrasion, bacteria enters into the nasal mucosa and causes the swelling of the face region and the false swelling nasal discharge. In the chronic cases, the involvement of the nasal and the facial bones occurs, which causes the permanent facial deformity. So the bull nose term is given, and its treatment includes the potentiated sulfonamides administered in the early infections. Thrush of the hoof. It is in the necrotic condition of the equine hoof, and it is associated with the poor hygiene, wet conditions, and the lack of the regular cleaning of the hoofs. These are the infections are the secondary to the hoof damage, results in the localized inflammatory response and are characterized by the wool spray. Treatment and control includes the regeneration of the frog by the providing the dry cleanliness conditions, regular attention to the hooves and the exercise. Black spot of the teeth, these are associated with the Fusobacterium necrophorum, and this causes the black box or the black spot of the teeth orifice and the sphincter of the dairy cow, and it is a localized area of the necrosis with black scab formation, and it may cause the stenosis of the sphincter and may lead to the mastitis. Coming to the genus Bacteroides, these are also non-spore forming anaerobic bacteria. These are pleomorphic or rod shaped, and they are normally non-motile except the two species. And these are non-capsulated, they stain irregularly, and the vacuoles may be present, and they occur singly or in the pairs and having the rounded ends. They grow on the blood agar and the Bacteroides bi-unsculin agar. College are circular, entire, low convex, translucent, and the semi-opaque, and less than 1% of the strain are the hemolytic, means the very less strains are hemolytic. And these are bioresistant. This characteristic distinguishes these genera from the other genera, and these can grow in 20% height. So this is an important characteristic, and these also ferment some of the sugars and the byproducts of the anaerobic metabolic are the some of the acids, and these consist of more than the 20 species, and of which the Bacteroides fragilis is the most important. And these are present in the intestinal tract and the mucous membrane of the human and the animals, and produce the bacteriocin, which inhibits the other bacteria. These are also associated with some of the important diseases like neonatal diarrhea in the foals, calves, piglets, osteomyelitis in the dogs, cats, horses, and also in the mastitis in the cows. These causes a wide range of the human diseases, including the abscess, diabetic foot, and the sepsis. And the enterotoxigenic Bacteroides fragilis are responsible for the watery diarrhea in the farm animals and in the young children. Dactylobacter nodosus, these are having rod shape with the terminal swelling, and these colonies are of three types, like papillate or beaded colony, mucoid colony, and the circular colony. Beaded colony or the papillate colony, they can be isolated from the fresh colony with having the advanced food rot condition. Where the mucoid type colony, they are present in the non-invasive infection of the interdigital skin of the sheep and the cattle, which are less pathogenic. And circular type of the colonies, they are present when the subcultured repeatedly in the liquid media, and these are avirulent type of colonies. And these are having the type 4 of the Fimbriae. These are non-flagellated, but having type 4 Fimbriae, and these provides a specific movement twitching motility, which is the flagella independent motility. And fimbrial proteins are highly immunogenic, and this is the basis for the serotyping. And these are some of the biochemical characteristics. catalase, oxidase, indole, urease, these are the negative, and the H2S is the positive, and it does not ferment the carbohydrate. It produces a thermostable proteases and elastase, and the having presence of agrolytic activity based on the agar-based media containing the powdered roof, and the type 4 fimbri is also responsible for the virulence, and also the basis for the classification into the 10 serogroups. This produces the foot rot conditions, and this may be progressive foot rot and benign type of the mild type of the foot rot, and these are present in the clawed and the hoofed animals, and along with the Fusobacterium, Diclobacter nodosus, and some other bacterial yeast are also responsible for the foot rot. And this is the highly contagious disease, especially in the case of sheep and the goat, and also in the case of cattle, horse, and the pigs and deer. They causes the lameness, severe weight loss, and if untreated, they may spread to the joints, bones, and the tendons. And as told, they, on the basis of the severity, these can be severe, benign, or the intermediate type of disease. Treatment and control, these includes the combination of the practices like vaccination, antibiotic treatment, food bathing, food trimming, and replacing on the flock, on the dry condition, keeping separate the healthy animals from the infected animals. So these concludes the topic for the anaerobic gram-negative anaerobes, and the important genus for these are Fusobacterium, Diclobacter, and the Bacteroides. So this concludes the topic.